Let's take a look at all 5 phases of mitosis a little bit closer. So the first phase of mitosis is called prophase. So pro in this case means first or before. So this is the first phase of mitosis. So during prophase, the chromosomes are going to condense or be packaged up so that we can actually see them, because before the chromosomes were all unraveled and you couldn't actually see the chromosomes at all. So it's just like when you're going to be moving things around, from one house to maybe you're moving to another house, you would put things in a suitcase or a box. So that's what the cell is going to be doing. So it's just going to be putting the chromosomes, that DNA, packaging it up, condensing it into a suitcase. So now we can go ahead and see them. And we've already copied or duplicated or replicated the chromosomes. So remember that each chromosome is going to consist of 2 identical sister chromatids. And then the nuclear envelope, the membrane that surrounds the nucleus where the DNA is also going to break down because that is going to be in the way. So we need to get rid of that nuclear envelope membrane. We're also going to have our mitotic spindle, which is also called the spindle apparatus, start to form. And the function of the mitotic spindle or spindle apparatus is that it's going to attach to the chromosomes and actually move the chromosomes around. So that's its function. So its function is to move the chromosomes around. So here's just a picture just showing that the cell is going to start off here in G2 of interphase. This is before it enters the M phase and starts dividing. And notice that we've got a membrane, a nuclear envelope around the DNA inside here. And notice that our DNA, our chromosomes in here are all unraveled, so they're kind of all spread out. So you can't actually see the individual chromosomes yet. But here we've entered into prophase, our first phase of mitosis. And notice that the chromosomes have condensed, so you can actually see the individual chromosomes now. So we've just packaged them up and put them into little suitcases. So that's how I like to think about these. Because if the DNA was all unraveled, like over here on the left, it would be pretty easy to break things and snag things. So it's just much easier to condense and package up the DNA and put them into suitcases, so when you're going to be moving around these chromosomes. And then also notice that the nuclear envelope, we've started to tear that apart, so it started to disintegrate because that's going to be in our way. And then we've also started to develop our mitotic spindle or spindle apparatus. And so what's going to happen is from these centrosomes in yellow here, they're going to send out these microtubules, these little protein sticks here, and those are going to eventually attach to the chromosomes and then move them around. All right, so our next stage of mitosis is called prometaphase. So the pro here means before metaphase, because metaphase is going to be our third stage. So the second stage is called prometaphase, so it just means the phase before metaphase. So in this stage, we're going to have these protein structures called kinetochores attached to the centromeres on the chromosomes. And kinetochores, I think of like suitcase handles. So kinetochores are suitcase handles, and they're just going to attach to the chromosomes and give you a place for the mitotic spindle or the spindle apparatus to attach. So let's go ahead and take a look at a picture of this. So here we've got our chromosome, with our two identical sister chromatids. In the middle here, it's being held together by the centromere. And then these tan areas, those are the kinetochores. So they're just made out of proteins, and these are like suitcase handles, because we're going to attach the mitotic spindle or the spindle apparatus to the chromosome, and we're going to be pulling on the chromosome and moving these chromosomes to the middle of the cell. And so when we're pulling on the chromosomes, we don't actually want to be pulling on the DNA that's in these sister chromatids. So we want to be pulling on the suitcase handles instead, these kinetochores. So that's where the mitotic spindle or the spindle apparatus is going to attach to instead, is to these kinetochores, these suitcase handles. Okay, and then our next phase of mitosis, our third phase, is metaphase. And meta means middle. So this one's an easy one to remember, because in metaphase, the mitotic spindle has moved the chromosomes so that they line up right in the middle of the cell. So that's metaphase. So metaphase, the chromosomes will be lined up right in the middle of the cell. Okay, and then our fourth phase of mitosis is called anaphase. And ana means apart. So what's going to happen in anaphase is that we're going to pull the sister chromatids apart. So you can see how the two sister chromatids get pulled apart, and we're going to pull them apart towards the opposite ends of the cell, towards these centrosomes, and the mitotic spindle just pulls them apart. And once the sister chromatids separate, they each become their own chromosome again. All right, and then our last phase of mitosis is called telophase, and telo means end. So this is our last phase, and cytokinesis is also going to occur during telophase as well. So during telophase, we're going to be forming two separate nuclei. And so our nuclear envelope, we're going to form a membrane around the chromosomes again. Our chromosomes are going to uncoil, we're going to unpackage our suitcases, and then our mitotic spindle will disappear because we no longer need it. And then cytokinesis is also going to be going on during telophase as well. And this is when we're going to divide the cytoplasm between the two daughter cells, and then finally separate the two cells so then they're their own individual cells. So here we've got telophase and cytokinesis. So you can see that we're forming a nucleus here and then another nucleus over here at the two ends of the cells, and our DNA is starting to uncoil and decondense, and our mitotic spindle is disappearing. And then you can also see that cytokinesis is happening. So we're starting to divide the cell right in the middle, so we can divide the cytoplasm between the two daughter cells, and then eventually this will go all the way through, and then we can separate the two daughter cells from each other. And the end result of mitosis that's really important to remember is we're going to end up with two genetically identical daughter cells. And so these cells will also be identical to the parent cell that we started with as well. So here's just a nice overview of mitosis. So here's the parent cell in interphase, and it's what we call a diploid cell, meaning it has two copies of each chromosome. And so the number of chromosomes that this cell would have would be 46 chromosomes. And remember that mitosis will happen in our somatic cells. So this just means any cell except the ones that are producing the egg and sperm. So The reproductive cells, they're the ones doing meiosis, but any of the other cells in our body will do mitosis. And so here are the 5 phases of mitosis. We have prophase, prometaphase, metaphase, anaphase, where we're going to separate the sister chromatids. They each become their own chromosome again. Here's telophase and cytokinesis. And at the end, we end up with 2 daughter cells that are genetically identical to each other and then also to the parent cell that we started with. So these 2 cells, since they're identical to the parent cell we started with, are also going to each have 46 chromosomes as well. Okay, so maybe the parent cell that we started with, maybe this was a skin cell. And then these 2 daughter cells would be genetically identical skin cells to each other and then also identical to the skin cell that we started with.